So let's have a look now at the citric acid cycle in more detail. So this is just an outline of all the citric acid cycle reactions. The first reaction involves acetyl-CoA. So remember, acetyl-CoA was formed by the decarboxylation of pyruvate via the pyruvate dehydrogenase complex. Now that 2-carbon acetyl group from acetyl-CoA is transferred to oxaloacetate to form citrate. Citrate has 6 carbons. So oxaloacetate therefore must have 4 carbons. Then the second group is an isomerization reaction. Citrate isomerized to isocitrate. Then there are 2 oxidative decarboxylation reactions in the citric acid cycle. The first is reaction 3, which forms alpha-ketoglutarate. How many carbons do you think alpha-ketoglutarate has? If this is a decarboxylation reaction, it must have 5. And then reaction 4 is the second oxidative decarboxylation reaction to form succinyl-CoA. So how many carbons must succinyl-CoA have? It must have 4. And that is the end of the first half of the cycle. We could consider where the objective of that is to create the 2 carbon dioxide molecules, or expel those 2 carbons that entered as the acetyl group out as carbon dioxide. Then the remaining reactions of the citric acid cycle are involved in reforming oxaloacetate. So the first one is a substrate level phosphorylation to form succinate. Then reaction 6 is an oxidation reaction to form fumarate, and this reaction uses FAD. Reaction 7 is a transhydration reaction to form malate. And then reaction 8 is malate being oxidized back to oxaloacetate. So carbon enters as acetyl-CoA and leaves as carbon dioxide. Metabolic energy is captured in the form of ATP. There's 1 ATP produced via a GTP actually in mammals. NADH, we said there are 3 NADH is produced, and FADH2, 1 FADH2 produced during the citric acid cycle. So basically what's happening here is acetate's entering the cycle and is leaving the cycle as carbon dioxide. So essentially that acetate carbon-carbon bond has to be broken so that 2 carbon dioxides can be released. What? So all we're really trying to do here is cleave this carbon-carbon bond of acetate. Do we really need this entire complicated cycle and all these other metabolites in order to get this to work? It seems like quite a lot of work. But the truth is that it is necessary, because in biological systems, the only way that a carbon-carbon bond can be cleaved, there are 2 different ways in which this can happen. Either there is a cleavage between an alpha carbon and the beta carbon of the carbonyl group. So remember, this is the carbonyl group, the C double bond O. The alpha carbon is the carbon next door, and then the one next door is the beta carbon. Cleavage can happen over there. The other way that cleavage can happen is it can happen between the carbonyl carbon and the alpha carbon, but only if the alpha carbon is attached to an OH group. So let's look at the acetate molecule, which is the structure over here. Do either of these apply? Are we looking at cleavage between alpha carbon and beta carbon? No, because there is no beta carbon. Acetate's only got 2 carbons. Do we have the situation where if we cleave between carbonyl carbon and the alpha carbon, there's an OH attached? Nope, there's no OH attached. So cleavage is not going to be feasible of this acetate moiety to form the 2 carbon dioxides as it is, and this is why the citric acid cycle is necessary, so that we can get that cleavage, so that we can get the carbon dioxides reduced. So what happens is the acetate is condensed with oxaloacetate to allow this beta cleavage reaction to happen. So these are all the reactions in the citric acid cycle. And just like with glycolysis, it gives the standard free energy as well as the free energy under physiological conditions. if we look at it, we can see there are three reactions that have the most highly negative delta Gs. They are the citrate synthase reaction, the isocitrate dehydrogenase reaction, and the alpha ketoglutarate dehydrogenase reaction. So what would you say is probably the case with these three reactions? They are going to be highly regulated. We also notice, wow, this reaction over here, the malate dehydrogenase reaction, is really not very favorable. And that is true, it is not very favorable, which means that the concentration of oxaloacetate present inside the mitochondria is actually very low. How does this reaction happen? Remember, it's a cycle. So this oxaloacetate is then used again in reaction 1. And we can see that reaction 1 is very favorable. And so what happens is reaction 1 actually pulls the oxaloacetate, the malate dehydrogenase reaction along. So the energy of this one is enough to pull the energy of this one forward, or that reaction forward. Okay, so let's have a look at the entire citric acid cycle then, starting from acetyl-CoA. Tricarboxylic acid. Why? Because citrate, or citric acid, is actually a tricarboxylic acid. You can see it over there. It's got 3 carboxylic acid groups, so that's where the name comes from. So the first reaction is a condensation reaction where acetyl coenzyme A and oxaloacetate combine via the enzyme citrate synthase. The next reaction is isomerization. Citrate gets changed to isocitrate via the enzyme aconitase. Then our next reaction is an oxidative decarboxylation reaction, and here we see isocitrate is converted to alpha ketoglutarate by isocitrate dehydrogenase. Oxidative decarboxylation, we can see NADH is produced as well as carbon dioxide. The second reaction, the second oxidative decarboxylation comes next. This is where alpha ketoglutarate is converted to succinyl-CoA by alpha ketoglutarate dehydrogenase complex. Also an oxidative decarboxylation, which means carbon dioxide is released, and so is NADH. The next step is a substrate level phosphorylation reaction where succinyl-CoA is converted to succinate, and in this reaction GTP is produced, which is then converted to ATP. This reaction is catalyzed by succinyl-CoA synthetase. Next reaction is an oxidation reaction catalyzed by succinate dehydrogenase, where succinate is converted to fumarate. And because it's an oxidation reaction, FAD is reduced to FADH2. Not NAD this time, but FAD. Next reaction is a hydration, so water is used to convert fumarate to malate. Then we have another oxidation reaction where malate is converted to oxaloacetate via malate dehydrogenase, and oxidation NADH is produced. Okay, next we can have a look at each of the reactions in the citric acid cycle in more detail. So the first is citrate synthase. Here, carbon atoms are introduced to the cycle in the form of the acetate moiety and acetyl-CoA. It's a condensation reaction where those 2 carbon acetyl group is added to the 4 carbon oxaloacetate. It is a ketone plus an ester. So if you have a look, oxaloacetate is a ketone and acetyl-CoA is a thioester. We can see that this is quite a favorable reaction with a very large negative delta G. You don't need to worry too much about the reaction mechanism. But what we can see is that the first part of the reaction, where the oxaloacetate goes to acetyl-CoA, has a K equilibrium constant close to 1, which means this large free energy that is comes from the second part of the reaction, where the citryl-CoA gets converted to citrate, and it really comes from the breaking of this thioester bond between the citryl group and the coenzyme A. So that's where the energy comes from. Of course, because it's such a large negative number, it is a highly regulated enzyme. And what regulates it? Obviously, things that are products of the citric acid cycle. So NADH obviously is going to down-regulate it. If there's a lot of NADH, it's going to want to inhibit citrate synthase. And similarly with succinyl-CoA, which is a metabolite, an intermediary metabolite in the cycle. So they inhibit citrate synthase, because when there's a lot of them, we don't need to make any more of them. Alright, so conitase is the next reaction, which is the conversion of citrate to isocitrate. Why do we need to have an isomerization reaction? Citrate's 6 carbon, isocitrate's a 6 carbon. Why can't we use citrate? Well, it's got to do with the oxidation. The fact that citrate is a tertiary alcohol, and we know that tertiary alcohols cannot be oxidized, whereas isocitrate is a secondary alcohol, and secondary alcohols can be oxidized. And we know that the next step in the pathway has to be an oxidative step. So oxidation is necessary. This isomerization process is a two-step process, where water is first removed and then water is re-added. So it goes from citrate to a conitate to isocitrate. In a very stereospecific reaction, where we are looking at this very specific pro-R hydrogen on the pro-R arm of citrate, and if you remember your chemistry, you'll know what pro-R means, is removed. Now, this OH that has to remove, because of course we are removing water, H and OH. This OH is not a very good leaving group on its own, and this is why this enzyme, aconitase, contains an iron sulfur cluster, and the iron sulfur cluster helps to remove the OH group. Then, of course, after a conitate is formed, the H and the OH of water are re-added now to form isocitrate, and we can see we go from a tertiary alcohol, so there's the OH group of the alcohol, and you can see that every single attachment to that carbon, not one of them is a hydrogen, so that is a tertiary alcohol, whereas here there's our alcohol, and we see there's one hydrogen attached, so it's a secondary alcohol, which means that isocitrate can be oxidized, because it's a secondary alcohol. And during the oxidation process, that hydrogen will be removed. So the iron sulfur cluster of aconitase, which amino acid do you think is involved here? If we're talking about sulfurs, it's the amino acid cysteine. So there are 3 irons and 4 sulfurs in the iron sulfur cluster. The sulfurs coming from cysteine. A 4th iron activates the enzyme. So this is what iron sulfur clusters look like. And the iron in the vacant position will coordinate the carboxyl and the OH group from the citrate. So it's this iron over here. The iron accepts an electron pair from the OH group, making the OH group a better leaving group. Alright, this is a question for you to attempt. Aconitase catalyzes. You're going to be getting these sorts of questions. When we do our tutorial, you're going to be having lots of practice with them, and you certainly will see them in tests and exams. Aconitase catalyzes the citrate to isocitrate reaction, and there's the standard delta G, but the delta G in pig heart mitochondria is 0.8. So we want to know under physiological conditions what